All right, so let's go ahead and get started. Questions on the study guide? Yes.
But it's still kind of confusing because parts of it, the sarcomere, like, what is that exactly? Like, is it within the muscle fiber itself, or is it a bunch of muscle fibers together?
Yeah, so a sarcomere is the contractile unit inside a muscle cell.
Okay.
So it's like almost the most zoomed in you can get.
So it's like an organelle, like, essentially. Like,
It's a structure, yeah.
Okay.
So like if you zoomed in, I got it actually up here. You start with the muscle and you keep zooming in, fascicule, muscle fiber. Myofibril is a whole bunch of sarcomeres.
Okay.
And then a sarcomere is the one little piece of actin and myosin.
The myofibril is what gives it the striations, right? Is that what, or is it
The sarcomere gives the striation.
Okay.
So if you put a bunch of sarcomeres together, end to end, you get a myofibril.
Okay.
And if you put a bunch of myofibrils together, then you get a cell.
Okay.
Can you explain the difference between the myosin and the actin? They just I kind of got the same difference.
Yeah, so the way to think of it is the actin is the piece that gets pulled on, and the myosin is the part that does the pulling. So the myosin does the pulling, the actin is what's getting pulled on. Yes?
Can you explain the isometric contraction?
Yeah, so the isometric versus isotonic, did anybody get those? Do you know which one's which?
Okay, the isotonic remains constant.
Yeah.
The isometric increase.
So which one involves like movement? Right? So like this is isotonic. Anything? If I'm just sitting here like this, that's isometric. But you can see I'm holding something, so there's still force. So it is a contraction, it's just nothing's moving. Right. So basically, yeah, pretty much always you're probably resisting gravity somehow. The isometric. So any contraction that doesn't produce movement is isometric. Any contraction where something moves, your fingers, your hands, your toes, that's isotonic. So I'm sure you've done any, exercising stuff. Isometric is usually the more painful kind. I think, like holding in a plank or, holding a 10-pound dumbbell out like this. Or like the shrug one. You've done that one. Anything else?
The A and the I bands.
So the A and the I bands. Show you a picture real quick, and we can talk about them at the same time. All right. So the I band is where you only see actin. So they kind of split the sarcomere, into little sections. And we'll talk more about it, but basically the I band is where you only see actin. There's no overlapping with myosin. So you only see the yellow. And if you look at it in the microscope, you can kind of see it there. So the I band is the actin. The A band, if you notice, it's where the actin and the myosin overlap and where the myosin is. Because up here is yellow is actin and red is the myosin fibers. So the A band is where the myosin and the actin overlap and where only myosin is. The I band is where only actin is. All right, so the answers. Which of these binds oxygen? Is it sarcolemma, T-tubule, sarcoplasmic reticulum, or myoglobin? It's myoglobin. Pretty easy. Myo means part of muscle. Globin means oxygen. Not a function of muscle tissue. It is all of the above. So stabilized body positions generate heat. How does your skeletal muscle generate heat? So the shivering, definitely. And moving substances on the body is also one. And what property of muscle is related to its ability to stretch without being damaged? Is it excitability, contractility, extensibility, or elasticity? It is extensibility. So they're very related, but they're a little different. So extensibility is you can stretch without breaking. Elasticity is you go back. You get pulled back to original shape afterwards. So rubber bands, for example, do they have extensibility or elasticity? They have both, right? So you can pull them and they don't break, so they're extensible. And then if you let go, they go back, which makes them elastic or have elasticity. So I want to make sure you got those. And what's the motor that performs contraction? Is it actin, titin, myosin, or Z-disc? It is myosin. So you're asking me, what myosin was. It does the pulling, which makes it the motor that's pulling. And what type of muscle contraction does not involve any movement? Isometric, isotonic? Isometric. So hopefully you got some of those. All right, so the reason this chapter is interesting and also hard is because you're going to learn exactly how, at the most tiniest molecular level, your muscles generate contraction. So instead of just saying, oh, your muscles contract and they get shorter, we get to actually learn how they do it and how it's controlled. So for part one today, we're basically just going to talk about what a sarcomere is, how it shortens, and then next time will be more about the signaling that controls contraction. So if you notice today, we don't talk anything about why it contracts or what tells it to. We're just going to stick with how it contracts for now. And that'll be basically the whole lesson. Of course, there's three kinds of muscle tissue: skeletal, cardiac, and smooth. Mostly just talk about skeletal muscle today. Cardiac muscle we won't talk about at all. Talk about that when we do the heart in 202, and just talk a little bit about smooth muscle. So of course, smooth muscle is the muscle that's involuntary, and you have it in your blood vessels, you have it in your intestines, it helps food movement. But mostly we'll talk about skeletal muscle. So which of these is not a function of the skeletal muscle? Everybody vote? Just want to make sure they're all working. All right, interesting. So everybody changed to D there, which is the answer. So that's smooth muscle. All the rest of those are skeletal. Because are your eyes moving? I know you're kind of thinking that's involuntary, but it's not. You are telling your eyes to look one way or the other. It's just not all that conscious, right? You don't have to say, eyes, turn left. It's more like you're thinking, I wonder what's over there. And they just kind of automatically move. But you are telling them to. It's just not all that conscious. All right. So as you zoom in more and more, you have these different levels. So when you dissected the chicken, you saw a muscle, or the bicep, the tricep. So muscle's made of a lot of fascicles. Fascicles are made of lots of muscle fibers, which are cells. And then the cells are made of lots of myofibrils. Myofibrils are made of lots of sarcomeres. So that's your list from complex to simple as you zoom in and zoom in and zoom in. What would be smaller than a sarcomere, do you think? To what? Actually, a nucleus is probably a little bigger. What are sarcomeres made of? Yeah, so if you wanted to go even one step farther, you could go sarcomere and then myosin and actin, which are just proteins. But we'll get those too. And so for today, we're basically just going to talk about sarcomeres. So hopefully you can get the idea. Let me turn off one of these lights. All right, so just to give you an idea of what it looks like as you zoom in. There's your muscle, and it's composed of a bunch of sort of cylindrical tubes full of all the muscle proteins. Those are your fascicles. And if you zoom into those, you start to see individual muscle cells. So you see how we're just kind of zooming in. And then if you zoom into the cells, eventually you get to a myofibril. And then if we zoom in even more, inside those myofibrils, which are these, you could see the individual sarcomeres. So from one Z disc to another is a sarcomere. And so you can kind of think of it as almost like just a parallel bundle of ropes over and over again. And so, the muscle would be the whole rope. Each little strand, like that'd be the fascicle, and then each little string in there would maybe be a myofibril. And then if you just took sections of that string, that would be your sarcomeres. So we're going to talk about what's happening here. And if you notice, everything's kind of attached lengthwise. So what do you think happens if this sarcomere gets shorter? Or I guess I should say, what happens if all those sarcomeres get shorter to the muscle? Does it get shorter or longer? It gets shorter too. So everything's happening here, but you just multiply it, by 1,000,000, and suddenly you get the whole muscle shortening. And of course, like everything, it is in 3 dimensions. So it is like big circles or big cylinders of sarcomeres put together. All right, so I'll just go through these a little bit, and then you're going to be drawing it, because I know there's lots of crazy vocabulary and structures up there. So this is a diagram. I have one muscle cell. So you've got in blue the cell membrane, and if you notice, there's these special proteins here, membrane proteins. They're going to attach to the cell next to it. So it's like those desmosomes or adherence junctions that we did way back in chapter whatever. So each cell is very firmly anchored to the other so that they move. When one contracts, it just pulls on the other one next to it. They don't like separate from each other. So all the cells are attached together. And we'll zoom into our myofibril. So this is one cell. It has lots of myofibrils, and the myofibrils have lots of sarcomeres. So if you zoom in to a sarcomere, there's a bunch of sarcomeres. Here's one sarcomere. So the basic structure of a sarcomere, in the middle, there's an M line. These are structural proteins that myosin attaches to. So myosin's in red here. So it's tightly attached there, which is good. You need something to attach to on that side. And then on the two ends, we have what's called a Z disc. These are more structural proteins, and that's actually what you're going to pull on. Attached to the Z disc is yellow actin, and then in red here is your myosin. And so when we contract, this myosin actually pulls on the actin, kind of like on a rope. And we're going to do a demo of that in a little bit. And you've got all your different zones. H zone is where you only have myosin. I band is where you only have actin from here to here. The A band includes the myosin and the myosin-actin overlap. So from here all the way over to there is your H zone. And then the last one, this titin here, if you notice it's attached all the way from the Z disc to the M line. So it's important for keeping that, letting it be extensible and elastic. It's that titin protein. Because if you can imagine, if you pulled your muscle, spread those Z discs apart, what would happen to the myosin-actin overlap? Would it go away, get bigger? What do you think? Imagine you're taking these two Z things and pulling them left and right. What would happen to the amount of overlap between yellow and red there? It would decrease. And if it became too much, like if it became none, then the myosin can never pull on the actin. But because titin's elastic, if you pulled it apart, the titin's going to pull everything back together. So it's like a little spring in there. And it's attached from Z to M. So if you pulled it, it's still attached, and you could pull everything back. So that's kind of how it works. All right. I had to fit a lot of instructions on this one. So basically the way I thought might work well for you to actually get an idea of what these sarcomeres look like is I've got step-by-step instructions for you to draw one on your table. So you're going to do it, let's see, all these instructions, in pairs, no books, and don't erase it because you're going to use it again in a little bit. So you'll need those markers, and I'll be walking around to make sure you're getting that. So I messed up on what I wrote on there for one little thing. So for E, it should say titin connects M line to Z disc. So I wrote it wrong on that paper. All right, so every sarcomere, you've got your two Z lines on the ends, and then the M line in the middle. How many myosins did I have you draw? 2 or 3? All right. So this is myosin. And then the actin comes about halfway from the other side. This green one's working. We'll make ourselves a Christmas one. Same thing on the other side. And then our titin comes from the Z to the M. You draw however you want. And then the different zones we're drawing. So the H zone is where you only have myosin. So it would start here and it would end basically here. That'd be the H. The A band's a little bigger. So the A band has all the myosin. So it would start a little over here, all the way to there. That's the A. And then anywhere where there's only actin, which is where the A band would end, that's the I. So I think most of y'all ended up with something like that. If you didn't, don't worry, it is pretty complicated. And of course, the reason to spend time looking at the structure is because anything else you do, if you don't understand how it's put together, none of the rest of it makes any sense. So the key thing to remember here is you've got the M line, that's what myosin's attached to. The Z disc, that's what actin's attached to. And then of course, it's not just one sarcomere in a cell, it's a whole long string of them. So then the next sarcomere would be exactly like this one, just right next to it. And then another one and another one, and so on and so forth. All right, so let's talk about how that structure we drew, how it makes your muscles actually move. So it's really simple. Basically, the myosin, this red part here, grabs on actin and pulls on it. And it kind of like this, like you're pulling on a rope, which we'll do in a second. And because actin's attached to the Z disc, if myosin pulls on all these ropes, what happens to these Z discs? They'll get closer together. And that's how your muscles actually make movement or contract, is these Z discs are getting pulled closer because myosin pulls on the actin. And basically they can keep pulling until the actin runs into the middle. That's how far they can contract. So myosin does the work, actin's like the rope attached to the Z disc. And myosin's the person pulling on it. And because all those Z discs are all attached together, and they're attached to the ends of the cells, and the ends of the cells are attached to each other, if you shorten all these sarcomeres, that means the whole cell shortens, and then the whole muscle shortens too, because it's all the cells are shortening at the same time. All right, so I think I won't make you redraw all of it. But next to your thing you already drew, why don't you draw it with contracted? But the only things you need to include, just put the Z, the M, the myosin, and the actin. So you don't need to draw all the bands or anything. All right, so you can see that up here, sort of what you were doing. So I think you already got a good answer there. Here's uncontracted, and you can see how far apart these two Z discs are, right, from here to here. And then down here, this is what happens fully contracted. So we moved all these Z discs closer together because we pulled on the ropes attached to them, which are the actin, basically. Do I? Yeah, so when it's contracted, if you look at this, The myosin and actin are basically completely overlapped.
So right now we're at H. So does each one like the actin will touch the M line and the myosin will touch the Z line?
So in maximally contracted muscle, yes. Actin would touch the M, myosin touches the Z, basically.
That little, the gray one makes so much more sense than the other one.
You like the gray one?
Yeah, it makes so much more sense with the I band and A band.
Oh, okay. I hate the gray one. But I'm glad we have both.
What part it really is.
Yeah, and now, of course, the reason they got all these names is way before we knew about what any of this stuff was. You could stain it and see, you could see this under the microscope, right? You saw these bands yesterday, at least some of them, right? You see kind of a dark and a light. That's what you were seeing. So they just gave them names because they didn't know what they were, really. Let's just call them A and I. That's why the letters don't really make any sense, right? Like H zone, what does that stand for? All right, so skip a few of these. I think y'all pretty much got it. Oh, we can do this one. All right, so I want to know which of those narrows or disappears when you're fully contracted. Let me put the answers up here, though. Answer choices. Where's the black one? All right. So A, H. All right. So which of those disappears when this is an uncontracted muscle. When it contracts, what goes away? H, I, M, or A band? All right, so why don't you talk to your group there and then we'll vote again. Wait, did I forget an answer? Oh, I need 2 more answers up there. Whoops. Sorry, I didn't put all the answer choices. Oh, I didn't realize it was 4. All right, 6 answers. Now you can choose what you want.
Right.
All right, so mostly people put E, which is correct. So if you look back at this one, if you squish them all together, what are you left with? Basically only overlap. And the overlap, do you remember what the overlap was? Yeah, the overlap is the A band.
So why does like the A band include the H zone?
So basically the A band includes all of the myosin, whereas the H zone is the myosin by itself. So if you don't have any overlap, you don't have any H zone anymore, and you don't have any I band anymore. Right. Let me do, yeah, all right. So another question. So which of these is true during contraction? All right, so chat with your group there. And if you do think it's... Do it? Oh, if you choose F, make sure you know which ones you think it is. All right, so I'll tell you that there's only one up there that is not true. So vote for the one you think is not true. So there's 3 that are true, 1 that's false. Or wait, how many are there? 5. 4 that are true, 1 that are false. 1 that's false. Choose the one that's not true. All right, so your time between, so most everybody's choosing C, which is correct. So if sarcomeres get shorter, Z discs come closer together, and then that's transmitted all through all the levels of the muscle. So muscle cell, myofibril, fascicles, the muscle. The tendon's the only part that doesn't change. But the muscle does, which is going to pull, on the tendon, which pulls on the bone. So they did get shorter to get you there. So for example, when I was holding this out, I had to get them shorter to get up here. But once I'm there, they're just staying contracted. Right. Mm-hmm. Right. They're just staying short. It's a good question. And it's a good point to your isotonic. It had to start somewhere. It didn't just all of a sudden be there. All right, so we'll do all the, I think I'll just do all about the myosin cycle next time. And the types of muscle and exercise, you'll just have to learn that one from these slides. It's pretty simple, but this contraction we'll definitely go over together. It's complicated.